Hello, I'm David, and today we're going to go through all of the ocular embryology you need to pass the FRCOphth Part 1 exam. So without further ado, let's get started. In the beginning, you have a zygote, which divides and divides until you get a morula where it's 16 cells, that's Latin for raspberries or something, and that carries on dividing until you get the blastocyst. Now, the blastocyst has two bits, a trophoblast, which becomes your placenta, we don't care about that, and the embryoblast, which becomes your human, and the embryoblast divides into two layers, the epiblast and the hypoblast. Since this is two layers, it gets called a bilaminar disc, but it's going to go on to become a trilaminar disc very shortly in probably the most important process in all of embryology, known as gastrulation. And that's when some of the epiblast cells flood into the middle and become mesoderm, at which point the layer on top gets called an ectoderm and the hypoblast becomes the endoderm. So ectoderm, mesoderm and endoderm forming a trilaminar disc. And it's called gastrulation because it also folds at the same time to form the bowels. Now the next process that's very important is neurulation. So I've got another slide on that. Neurulation is the formation of the central nervous system, and basically the surface ectoderm here folds into a little tube which pinches off from the surface ectoderm and becomes the notochord, which forms your spinal cord and your brain. Now it forms in the middle here where it closes up, and then it closes up first at the anterior neuropore at day 24, and finally it closes at the posterior neuropore here at day 26. Failure to do either of those things will result in a neural tube defect, anencephaly if that end, and spina bifida if that ends. We don't like that. But the most important bit on this slide is that once this notochord is formed, at the crest of it, the cells called neural crest cells come off and they migrate all around the body and form loads of random structures. To summarise the layers then, you have surface ectoderm, neuroectoderm, neural crest cells, mesoderm and endoderm. So yeah, the first three come from the same layer of ectoderm, but once neurulation happens, you've then got neuroectoderm forming the notochord and those neural crest cells which migrate around the body. Now we can talk about the formation of the eye itself. So the primitive brain has three parts, the forebrain or prosencephalon, the midbrain, the mesencephalon, and the hindbrain, the diencephalon. And in the prosencephalon, the forebrain, it folds, forming this optic vesicle here. And eventually it folds so much that a little bit pinches off from that surface ectoderm to form the lens. Okay, so the origin of the lens and the corneal epithelium here is surface ectoderm, not neuroectoderm, which is this bit here. Okay, all of this stuff here is neuroectoderm with the surface ectoderm covering the whole embryo. Now, this happens at day 33, so you need to remember that day. Day 33. And when it fails, you get a family of anterior segment dysgeneses called keratoiridulo-lenticular dysgeneses, cornea, iris, lens. The most common of which is this, which is called Peters anomaly. So you can see this adhesion between the cornea and the lens. So there's missing endothelium here. There's also adhesions to the collarette of the iris here, and sometimes you even get adhesions to the lens, which can then turn into a full-blown cataracts. So you get type 1 Peters anomaly, where the lens isn't involved, and you get type 2 Peters anomaly when the lens is involved, and it's usually bilateral and is actually more common. About 60% of Peters anomaly is type 2. There are other keratoiridulo-lenticular dysgeneses as well, but this is pretty much the only one you'll hear of. So this is what happens when that lens vesicle fails to pinch off completely from the surface ectoderm. Now, assuming the lens does form properly, you might be thinking, well then how is it nourished? And the answer is the hyaloid vasculature. The main bit is the hyaloid artery here, but there's other bits. So labelled number 1 here is the tunica vasculosa lentis, which is a coat of blood vessels surrounding the lens. Then you've got the hyaloid artery, and then number 3 is the glial tissue surrounding the back. Now, you want the vitreous to be nice and clear. You want the lens to be nice and clear. So all of this has to regress. It has to disappear by the time you've got the adult eye. When it doesn't, you get what's known as persistent fetal vasculature, which used to be called persistent hyperplastic primary vitreous before it was recognised that this is a misnomer because it can affect the anterior segment of the eye as well. Now, when someone calls something PFE. What they're probably talking about is full-blown PFE, where the whole of the hyaloid vasculature remains, forming this fibroblastic core of tissue going from the lens to the retina. And often the retina is dysplastic and even detached and folded in with this tuft of tissue going all the way through the vitreous. These eyes are pretty much always inoperable, because if you've got this, then the cornea is usually affected, the optic nerve is affected, everything's messed up. So when people say PFE, they usually mean this, but there are other forms of PFE which are minor and often thought of as benign conditions, which you just see from time to time and don't really have to do anything about. And these are the main three. The most common is probably a persistent pupillary membrane. This is a very serious form of it. It's a remnant of the tunica vasculosa lentis, and the most common form, you'll probably just see a tiny strand of iris going across from one bit to the other across the lens, and that's fairly common to see in the average person. The other two forms of PFE that you might see more regularly than this full-blown form is this. This is when you've got a dot of hyaloid vessel remaining on the back of the lens. It's called a Mittendorf dot, and it can lead to a cataract. And this is when a bit of hyaloid vessel remains on the optic disc, and this is called a Bergmeister papillae. You don't really need to do anything about these, unless you want to do cataract surgery for this, of course. The only other thing worth saying about the lens is that the lens epithelial cells form in sutures, so you get a Y-shaped suture anteriorly and an inverted Y posteriorly. And the reason why it's worth knowing this is because you can get a sutural cataract, which is what this is. This is when you get a opacification of a suture. It's usually a congenital condition, often unilateral, and because this Y-shaped one is anterior, it often doesn't even affect the vision much and might not even require surgery to prevent amblyopia. Now, continuing on with the development of the rest of the anterior segment, pretty much the rest of it is formed from neural crest cells, and there are three migrations. At day 39, you get migration 1, which forms the corneal endothelium. Don't worry about these days, by the way, they're not going to ask you about these. The second migration forms the iris, pretty much, and the third migration forms the corneal stroma, so it floods in between the epithelium and then the endothelium, forming this stroma here. So those are all formed from neural crest cells. And when it goes wrong, when those migrations don't happen properly, you get what's known as an iridotrabecular dysgenesis, which is a whole family of disorders, the most basic of which is called posterior embryotoxin. Posterior embryotoxin is a prominent and anteriorly displaced Schwalbe's line, which looks like a white line, and it happens in about 6% of the population, so it can be a completely benign finding you don't need to do anything about, but it's also associated with certain systemic conditions such as Alagille's disease. Now, when you get posterior embryotoxin plus iridotrabecular adhesions like these, it then becomes known as Axenfeld anomaly. When you get this plus iris dystrophy, it then becomes known as Axenfeld-Rieger anomaly. So these two things are often known as an Axenfeld-Rieger spectrum. Now, you can get Axenfeld-Rieger anomaly as part of a systemic condition called Rieger syndrome, which is when you get an Axenfeld-Rieger anomaly plus microdontia plus hypodontia plus redundant periumbilical skin. So you just get a big flap of skin around the belly button, basically. They can ask you about that in the exam, believe it or not. I know that's a lot of names, but just go over it again. It's fairly easy to remember. The last of these iridotrabecular dysgeneses is one you'll definitely have heard of. It's called aniridia. And what I want you to notice is that even though the name suggests there's no iris at all, in most cases you do get bits of iris, so it's not a complete aniridia. It's associated with the PAX6 gene, and you want to learn all about Wagner syndrome, where it's associated with nephroblastoma, and you might need to do an ultrasound scan, and they can ask you about that. So those are iridotrabecular dysgeneses when these neural crest cell migrations don't happen properly. So that's the formation of the anterior segment. Let's talk about the posterior segment. This is fairly easy to remember. It's just, well, the neuroectoderm folding, you get that intraretinal space which flattens out, and you get the choroid, which is the non-sensory retina, and you've got the neurosensory retina here, which becomes the retina that everyone knows about. That's all there is to it, really. As for the ciliary body and the iris, basically that's from this bit of folding. So the anterior ciliary body and iris forms from where this forms from the same layer as the choroid, and the posterior bit is formed from the same layer as the retina, which I have another slide to show you. So there you go, the pigmented layer of the retina, from which the choroid forms is the same layer from which the ciliary process forms. And you've got the neural layer of the retina here, which will form the actual neurosensory retina. Now, the last thing you need to know is that this optic stalk has a fissure in it through which the hyaloid vasculature runs, and this choroid fissure, as it's known, closes, and it closes on day 33. You should remember that the other day I told you to remember was also day 33. So if you get asked to name a day, just put 33 and you'll probably get it right. So why is the choroid fissure so important? Well, that's because when it doesn't close, you get this, a coloboma. And a coloboma is a gap which can extend all the way from the iris all the way to the optic nerve. So here is an iris coloboma, and here is a coloboma involving the retina, the choroid, and the optic nerve. And the giveaway is that it's always inferior nasal because the choroid fissure is inferior nasal. So obviously there's lots of retinal conditions that can look a bit like this, but one of the big giveaways is that this is inferior nasal. So that's why you need to know about the choroid fissure. So there you go, that's all the ocular embryology you need to know. This is a nice summary slide of where all the structures of the eye originate from, because probably the favourite question they like to ask when it comes to embryology is which of the layers does structure X come from? So surface ectoderm, you'll remember, is lens and corneal epithelium, but it's also worth knowing that the skin of the eyelids and the lacrimal glands, which is sort of buried in the skin, also comes from the surface ectoderm, which is just the same layer that the skin of the rest of your body comes from. Notice that it says vitreous. They're not going to ask you about the origin of the vitreous because the development of the vitreous is very complex. You have primary, secondary and tertiary vitreous, which all come from different places. And I've not told you about that because you just don't need to know about it. Then you've got neuroectoderm, which is the layer from which your central nervous system comes. You've got retina, the optic stalk, and the epithelium of the iris and the ciliary body. Also note the muscles of the iris. Remember I said that the neuroectoderm folds at the end like that, so that's why that's that. And then you've got the mesoderm. Now, as you might remember from medical school embryology, the mesoderm is the layer from which all the muscles and bones come from. That originally starts as those somites, which split off into segments. So the extraocular muscles come from mesoderm. Endothelium of the blood vessels comes from mesoderm. The sclera is another one they're unlikely to ask you about because the temporal bit comes from mesoderm, but the rest of the sclera comes from neural crest cell migration. And then again, we've got the annoying vitreous, which we don't care about in terms of embryology. And then lastly, we've got the neural crest cells, which told you about the corneal stroma and endothelium, most of the sclera. Just think about the cornea and lump it in with that category. And then trabecular meshwork, melanocytes, all melanocytes in the body come from neural crest cells, and the ciliary muscles and the ciliary ganglion. So there you go, that's everything you need to know about ocular embryology for your Part 1 exam, and probably the rest of your life. Thanks for watching.